Recording in progress.
Okay, thank you, Slesa. So you can now move on to NCBI and download any of your gene of interest. And if you are not familiar with these things, then you can directly type ACN9A. ACN9A. So you can just add nucleotide here. And you can sort this. So those who have completed this, go on to NCBI and then type SCA, SC9A gene. It is a gene for congenital insensitivity to pain. So interesting gene. I again request everyone to please remind us that if you are done with downloading the things, please inform us so we can move forward, because we are just running out of time. We need to finish it within this 2 sessions. So later on we can do all the thing as well. So please, if you are having any kind of trouble, please inform us. We will be helping that. Should we download the gene sequence? Yeah, you need to download. You do not need to download. You just need to open up that. Please wait for me. So I'll be guiding you there. You can also download the sequence and then move on to another program, but it's very simple there. So just follow me. So maybe all of you have downloaded the sequence, downloaded the tools. So once you have downloaded, you can move on to database, NCBI database, and you need to mention the nucleotide or gene or something like that. And then you need to type on one particular gene. If you are familiar with different genes, then you can directly type on the name of gene so that you can get the result there. Or if you are not familiar with those things, then you can type SCN9A, which is already I've mentioned it. It's a gene related to congenital insensitivity to pain. So person do not feel any kind of pain in this disorder. So once you type this, you need to sort it. So, and the results appear like that. SC9A, sodium voltage get whatever the thing you just forget about everything. You just go on through doing the things. So later on, once you do all of the things, then you will be analyzing all the sequences, and you will be going on to other perfection. So for now, you just go through the procedure. So it's SCN9A, sodium voltage gate channel alpha sub units. So you can directly do blast from here as well, or you can download and move on to next step. I'm just doing blast from here. I'm not selecting any other. For example, there has been obtained a lot of gene over there. So, but I'm just selecting this one, sodium voltage gated channel. So if I do blast from here, then it will directly give me a lot of sequences that are closely related with this particular gene. Let's do blast from here. So it's a blast platform. It's a blast home page. So there's blast in, there's blast P, blast EX, and other thing as well. So we are getting the nucleotide sequence, DNA sequence. So for now, I'm doing blast in. It will be analyzing the sequence similarity between DNA and DNA with some gaps. So I'm doing these things. So you can see the accession number given there, NM underscore. These are things, and then you can see other things there as well. So program selection, you can optimize the program. It is already in the default form, highly similar sequences. So it will, it means that it will give the result of different sequences that are highly similar to this parent sequence. And there are a lot of other algorithm parameters that you can set up there. So just go on and do blast there. Let's do blast here. So it will take some couple of seconds. It will not take more than 5-6 seconds. So you'll get a result there. If your protein, if your sequence is too large, then it will take some minutes. Otherwise, it's just a piece of cake to pop on these things. So here we go. We got a lot of similar sequences regarding to that particular SCN9A gene. So here is one gene that is a highly similar sequences. This is a number, organism that is obtained from, and all the things. I'm not going to e-value and all the things. You will be later on analyzing all of things, because if I go with this e-value, and in case of him tree, if I go on to bootstrap and a lot of things, you'll get confused. So we'll not go through that. Once you do all of the things, then in competent training or in workshop, we'll be analyzing all of the things, values, and all other post sequence analysis, even in gene integration and a lot of things. So now we are just doing multiple sequence alignment and phylogenetic analysis of this particular sequences. So you can also directly do multiple sequence alignment from here, MSA viewer, and then you can also analyze the phylogeneticity from here as well. You just need to select the particular sequences, 2 or 3 or maybe 4 or multiple sequences. You can select multiple sequences from here, and you can directly move on to MSA viewer, and then do multiple sequence alignment, and then you can go on to perform phylogenetic analysis as well. So that is the step from online tool. You can also move on to UniProt, and you can do those things, but that is not so much given emphasis to your people. So just follow the online tools that has been asked to download, and then we will move on to those things. So let's select some closely related sequences. So I'm going to unselect all of the things, and I'm going to select 2 or 3. Okay, let's do 3. So this is from Homo sapiens. It is from something like that. So let's select these things, and we can directly move on. So you can also directly download from here, or you can just select one and then extract a FASTA format, and then you can copy those sequences into your notepad, and then later move to cluster, and then to phylogenetic analysis. So let's do it individually. So let's click on this particular gene. If I click onto this particular gene, I'll be getting this result. So it's our information obtained from this particular gene, the first similar sequences. So here we go, the fastest sequence of this particular gene is closely related. if I click on to FASTA, then I'll be getting FASTA sequence here. You need to copy this FASTA sequence into your into a notepad, and then you need to copy it. But remember, this greater than sign should be there because it is a FASTA sequences, FASTA file format. Sorry, FASTA file format. It is literal, that is a text-based format which is used to represent DNA sequences, and these all are the single letter representation of amino acids. For example, ACGTN. That means A for adenine, and C for cytosine, G for guanine, T for thymidine, and so on. You all know that basic concept. So you just need to copy this FASTA sequences to your notepad. So for copying it, I just copied this one. Just follow this. So I've already copied it. So I'm just guiding you people. So just need to copy this and then paste into notepad. So I've already done this. I've mentioned the name of that particular gene as 1. You can mention anything else. So please give on to this sign. Otherwise, the cluster X it will not be recognizing this FASTA sequences. So this is the first sequence. So that has been done. You need to move on to next one now. This is the next sequences. We are going to do the sequence alignment. So you need to click on this. You'll be getting the second sequence here. Move on to FASTA. You're getting FASTA sequence there. And then similar to the previous one, prior one, you just need to copy this FASTA sequences. I've already copied it. I can directly show you there. So similar to the first one. So it's a second gene. Okay. If you have any problem, please ask me. Maybe I'm not going too fast. So it's a second gene. So similarly, you can copy the sequences, third sequence as well. This one is second one, and then this one XM 0169495458. That's the next sequence, third sequence. So you just need to copy this third sequence. FASTA. You just need to get that FASTA sequence. This is all about the third. That's we are done now. This is the third sequence obtained here. So I've given the name 3 for that third sequence, and that's the FASTA file format of third. So in as a whole, we have copied 3 FASTA sequences from a database. So now we will be moving on to multiple sequence alignment. If you have any question, please. Maybe you all have done this up to this step. If you are done, then please inform, or if you have any kind of problem, please let us know. Okay, now, so it's a software informer. I've already shared the link. So if you type on cluster X here, then you'll be getting this result, and you need to download this cluster X program for multiple sequence variety. It's a very basic tool, and it has been already changed into Omega, but cluster Omega, but this cluster X is user friendly, so it's very simple as well. So I'm doing through this program. Okay, you just need to download this, and then you will be extracting. You'll be extracting, and you can get this result. I've already copied this application here onto the taskbar. You can see this. You need to open this now. Oh, maybe screen is paused now. Okay, let me share it again. So you will be getting this one. If you look at the screen right now, then I'm clicking onto the cluster X. I've already drag this to my taskbar, so I can directly move on to cluster X. You can go to the source bar and then you can type on cluster X and you'll get that application, no matter where you have code up to it. So let's go on to cluster X now. So it's a cluster X. So we have already copied 3 sequences there. So we have already copied this sequences, this file from this file. So I have put this text, this notepad document here. So later on, I'll be going on to training folder so that I can directly get access to that particular sequences. So you can do like that. You can create a one file format and you can directly move on to there and do the thing. So let's move on to cluster X now. So after opening this home page, you just need to go to file. You just need to go to file and then load sequence. Load sequence, and you need to move on to that particular file folder. So it's a text format. You can just open this. So it's a basic thing you need to do here. So we have already opened this theme now. So now you need to move on to alignment. First of all, you go to file, you load the sequence. You need to go to a particular folder where you have kept the 3 sequences in one single notepad. Then you do the alignment thing. Do alignment. That's the thing. So that's the thing you need to do. Now, how did you get all the sequence at once? Okay, I'm not getting the sequence once. I've also already showed you. Please select one single sequence and then paste onto your text document, and then copy another sequence and then paste there, and then you will get a multiple sequence there. Maybe you have not followed my steps before. Please go on to the previous one. You go to NCBI, you just type that gene name, and then you'll get and then you need to do the blast, and then you get many of the sequences there, many of the close related sequence. You just need to pick 2 or 3 or 4, maybe that depends upon you, and then you copy one sequence at a time. Not all, do not copy all of those things, and there is no available as well. So you just need to click on one, you select one sequence, and then copy that sequence to your text folder, notepad, and then another one and another one, and so on. I have to copy all the sequences in the same file. Yeah, you need to copy different 3 or 4 or something like that to a single text folder, so that the cluster X, it will be recognizing all of the sequence at one single folder. And just remember that you need to put that greater than sign once before the sequence, right? So that, and the cluster X, it will be recognizing that sequence. That's it. Hope you have done that. Now it's
Hello,
all about
excuse me. Hello,
yeah?
Excuse me. Please, how do we paste the sequence? Do we separate them? Is there a way to separate them?
You just need to copy that to your notepad. You just create one notepad folder there, and then you copy that FASTA sequence from the database and then paste it here.
Is there a way to separate them, maybe a space or a gap or a line? No.
Excuse me, I didn't get your point.
How do you differentiate this? Okay, you put a greater mark and 1 to separate all of them.
No, not in a separate folder. You just copy one single sequence and then paste onto your notepad folder, and then another one and another one. That's the step. Do
So
it individually. Do not. Yeah.
I'm asking, is there a way to separate them to show that they are different sequence, like a comma, a full stop?
Yeah. Once you do the blast sorts, then you'll get the result. So all of these sequences are different. All of these
I
sequences are different.
I know they are different. My question is, when pasting it on the notepad, is there a way to separate the sequence to show that this is a different one, this is a different one? Like I see you have a.
Okay, I got it now. So you can go on to your notepad and then indicate your own indication, 1 or 2 or 3 like that, so that you can separate those sequences. For example, in case of mine, let me show you. In case of mine, I have separated the 3 sequences by 1, 2, and 3. So you can name your sequence yourself.
Oh, okay.
For example, 1, and I have created NS sequences as 2 and then 3 there. Got it?
Thank you. Yeah, exactly.
Okay. Thanks for the queries. So let's move on to the cluster X now. So once you do this, you need to move on to alignment and then align the sequence. And first of all, you need to create the output file format as well. So click onto this. There is MSA file format. So cluster X, it produces MSA file format as an output result of multi-sequence alignment. So you need to click on here, and PHYLIP, it's another file format for multi-sequence alignment. So you need to click that as well. So once you're done, you just need to click on this. So now you need to do complete alignment. So you can create your own target folder. So in my case, I created one training folder. So all of the file format, DNV and PSY and MSA file format, that will be extracted into that particular file folder. So that's it. You can create your own here. You can go to browse and create any target file folder there. So once you do this, you just need to do OK. So it will be operating by itself. So you can see the result here. Align is cool, 99%, sequence is 1, 3, and so on. Once it has been complete, then all of the file formats, the extracted file formats, that will be downloaded to the targeted folder. That means in my case, it's a training. So as a result, I'll be getting different file format, DNP and PSYLP in that particular file folder. So the alignment has been done. So let's move on to the analysis of the results. So if I move on to training, then I now can get different file DND, and this one, this is for GeneDoc. In GeneDoc, we'll be doing the result will be analyzing there. So that's the thing. So once you do this, you need to open GeneDoc now. So I've already pinned my GeneDoc app there into my taskbar. So I just click on this and I'll be going there. So just go to GeneDoc. So once you go to GeneDoc, then you'll be getting file project and a lot of things there. So you just need to go to file and then open. File and then open. So you need to go to training. You can get this TXT, that is GeneDoc file folder. So you need to open this one. Once you open, and then you will get the result. This is a multiple sequence alignment result. So you can see there the sequence differences and conserved region as well. So you can see there is a difference in amino acid base pairs. and you can see a conserved region here. So in case of analyzing this conserved region, it's a result for analyzing this active site as well. It's a nucleotides. So if you are doing the protein, then you can directly say that it's an active site in that protein. So in case of protein, you will be getting amino acid into that. So these are the amino acid conserved region, and in between this, you can see the active site. So in molecular docking, we'll be performing our ligand will be performing molecular docking in this particular active site. So you can see a lot of sequence similarity here. So it can be considered as active site. So you need to compare this active site with database as well. So later on tomorrow we will be doing that. That's it for multiple sequence alignment. Maybe you all have get the result. Now let's do phylogenetic analysis. So in case of phylogenetic analysis, you need to download that tree view X. So I've pinned it as well. So I'm opening this tree view. You need to just open this tree view. So in case of tree view, you need to copy this DND file format. This for phylogenetic analysis. So just drag this file format to tree view. So you can get the result as simple as it is. So you can see 1, 2, and 3 sequences. They have a common ancestor. That is a analysis of multiple. That is an analysis of this phylogenetic tree. So it's a phylogenetic tree. 1, 2, and 3 are clades. So these 3 clades have a common ancestor. So if you are analyzing a lot of sequence, then you can get multiple clades here. Then you can analyze the result. So that's a basic thing. We are done with this thing. If you have any kind of trouble doing this and all the things, you can ask me. We have just 7 minutes left, so we'll be closing this session later on. Tomorrow we'll be joining and continuing the session. If you have any problem, you can ask.
Hello.
Yeah, please.
Yeah, please, can you show how to run the class again?
Can you repeat here?
How to run it on the cluster X.
How to? I didn't get you.
I got stuck where you after copying the sequence and pasting it on the notepad, so you ran it cluster X. I kind of skipped that. I didn't get how you did it, please.
In case of multiple sequence alignment, you're talking about multiple sequence alignment?
Yeah, can you show how to use it on the cluster X software?
Okay, so in case of multiple sequence alignment, you just once you create that notepad and you copy all the sequences, maybe one or more than one, at least you need to select 2 sequences, more than 1 sequences, 2 or 3 or 4 or 5, doesn't matter. So you can look at this notepad. I created 3.1, so it's a notepad. It's a file format that I've created in a training folder. So you need to go to file. Just follow me. You need to go to file. You need to load that sequence.
Okay.
Got it? You need to load that sequence. And then you will be finding this result. Okay? Okay. Then you need to go to alignment. Got it?
Yeah.
Okay. And you need to go to alignment and you need to go to output file option. In output file option, you need to mark this MSA file format and PHY LIP file format, and then you need to select. You need to do that, and then you will be going to alignment, and you need to do complete alignment. There, got it?
Yeah.
Okay. Once you do the complete alignment, you will be getting number of file formats, and then you need to open this gene doc. Okay, got it?
Yeah.
Okay, once you open this gene doc, you need to go to file. You need to go to file and open. Got it?
Yeah.
And open and then this file format, this program will directly redirect you to your target folder. So it's my target folder, and I've already got that txt document from ClusterX. So you just need to click this one. Okay.
Yeah.
Okay. Once you click this, then you will get this result. This is a multiple sequence alignment result. Got it?
Yeah.
Okay, that's it.
Okay, thank you.
Anyone, if you have any queries, if you have any kind of trouble, then please inform us. We are running out of time. We'll not be doing another session. Tomorrow we'll be joining, and we will be doing it tomorrow. So we have just 3 minutes left. And if you have any queries, or if you have any questions regarding the session, then you can just forward it. And if you have not registered, then please go to that Google form and please get it registered, so that we can send some kind of this visualization to link over email, so that you will be downloading those visualization tools, and tomorrow we'll be practicing there as well. If anybody is having trouble with these things, then please inform us. You can just send a text there. And you can also communicate with us in Facebook or WhatsApp, or even you can send an email if you have any queries, so that we will be happy to assist you there. So we've got one question. Would you like to send me this software link again? Okay, okay, I will send you it. Okay, we have less than 1 minute, so maybe you have all understood everything. Hope to be so. If you have any kind of trouble, then we can assist you tomorrow. So please do practice all the things that we have shared the information there. So once you come back tomorrow, then it will be easier for us to assist you as well. So we'll be moving on to tomorrow's session, and if everything goes fine, then we'll be doing molecular visualization and some kind of simple molecular modeling in Chimera and other visualization tools tomorrow. So thank you for your presence there. Hope it went fruitful for you all.